Hello, today I will teach you a very important bioinformatics tool that is useful for quality control and pre-processing of sequencing data. This is fastp. This tool is used either you are working with single-end or per-end sequencing alignment. So let me tell you what fastp and why fastp matters. So when we receive the raw sequencing reads from the sequencer, they frequently come with low quality bases, adapter contamination, and sequencing artifacts. These must be corrected before we proceed with any downstream analysis. That's why a FASTP is a tool that is able to do the quality filtering that we need, adapter trimming, length filtering, base correction, poly-G trimming. And besides that, we can easily generate a report that can be in HTML or JSON format. So before moving forward, let's talk about the bioinformatics pipeline. After receiving the raw read, we need to do the quality control, trimming, and filtering. This is what FASTP is doing with us or for us. So when we have this job done with FASTP, our data, they are ready for any RNA-seq analysis, variant calling, genome assembly, and metagenomics. So what do we need to perform FASTP analysis with FASTP? We do need a terminal environment. That's required. But the computer environment like a Windows, Mac, a Linux, or Mac OS, that's depending on what you have, but what is required is the terminal environment. So for Windows, I do recommend the WSL Windows subsystem for Linux. And we also need to install the FASTP. To install the FASTP, to ensure that we have a good reproducibility, I do recommend the use of Conda, that is the open source package manager. So after that, we need the files to process. If we are doing the single end alignment analysis, we just need one file. But if we are doing the pair end analysis, alignment sequences, we need the forward sequencing data and reverse sequencing data. That means we need two files. We do need some disk space based on what you are, what kind of data you are using. The disk space requirement may vary. So before getting started, let me let me teach you how to use the Conda, how to install FASTP, first of all. To install FASTP, there is several ways you can install FASTP. You can install it. There is an official GitHub page where you can use to install the binary version of FASTP. You can also use it directly in the command line using the sudo apt, but that sudo apt requires a admin credentials. That means it's not good for reproducibility. For reproducibility, I do recommend Conda because with Conda, you can share your codes with anybody. What you need to do is just install the environment. You don't need any admin credential to use the Conda environment. Let's try to do it. If we use the Conda create -n fastp environment, this is the name for the environment that you are creating. It can be other things. It's not need to be exactly fastp environment. But what is required is Conda create -n, the name of the environment, and -c bioconda fastp. I will not run the first line. Why? Because I already have the Conda environment activated, but I will just activate it to be able to work with you. Since we have it activated, since you have it installed, sorry, you need to activate it. Because I already have it installed, I will just activate it. So I don't need the first line, but if you haven't used the Conda environment first, the package manager, you do need to use the first line to install the fastp. So let's do Conda activate fastp environment. I think I mistyped it, fastp. Okay, fastp environment. Okay, let's see what is changing. Sometimes it's taking maybe 1-2 minutes. It's depend of the speed of your computer. You see my environment is activated. So now I am ready to work. So before we proceed with the fastp analysis, we need to have the data. For the data, I do have some data I found on the OpenGene GitHub account. That is a open, a publicly available. I use the small set of data for the purpose of this tutorial. So we need to clone the repository. You can use git clone, just the URL of the repository. Let me just type it because it is not that long. But if it is long, I do require to copy and replace it. github.com/openGene/fastp.get. I hope I didn't mistype nothing. Okay, now it's cloning. I didn't mistype. That's good. Okay, so we have the repository clone. I think what we need to do, and the last part, the fastp.git, the fastp, that means this is the folder that is created in my, now I'm in the home directory. That means in the home directory, I should have this folder. Let's check if we have it. Yeah, the folder name fastp. Yeah, we have the fastp. We have the fastp, for that means we successfully clone the repository. So we can go inside the fastp to see what is inside. You see license with me. What we need, we need the data. They're supposed to be in the test data because this is for script. Yeah, this is supposed to be. Let's go inside to see test data. Voila. Yeah, we have the forward data and the refresh alignment data. So let's move back to the home directory. Okay, now since we confirm that we have downloaded the data from the GitHub, now what we need our directory for this project. Let's make our directory, make directory. I will put this flag -p because if the directory already exists, it will just silently create. If it doesn't exist, it will create it for you. -p. Let's call this directory fastp tutorial. Yeah. Okay, let's move inside this directory, or if you want, if we can go into that. Okay, let's just copy it's the file we have and the fastp directory that we just downloaded. Just cp, that's for copy. We go for the file we have. Okay, fastp. We go fastp test data. This is where we confirm that we have this is where we confirm that we have the data when we do the test data. And this line, we can just reconfirm that. And I will go in this line, we have the R1, R2, that they have almost the same format. I will just the wildcard asterisk to copy everything that end with .fastq or FQ. Where I am copying that, to the directory that I created, that is fastp underscore tutorial. Okay, now let's move to this directory to see if we successfully copied everything. Let ls. Voila, we got the R1 and R2. Okay, we can move for the next step. Okay, now this is just to understand the FASTQ format. This is just the how to understand. Let's just open one of them to just compare. We have the three until 4 lines. The first line is just the unique read name. We have the DNA sequence, the separator that's usually a plus sign, and the quality score. Let's just open just cat R1. Voila. This is technically what I was just talking about. This is the first line. This is the root identifier. This is the DNA sequence. And this is the separator. And this is the fourth line. That is the quality score. Yeah, let's create because it's start getting messy just to get more space to work. Okay. Okay, now let's proceed to do a single end read analysis. Let's run in fastp for the single end. But before doing that, I forgot to tell you that I do recommend to zip or to compress the file because when you compress the file, specifically when we are working with file that is big in size, it's better to move them because you can it can take time to copy or to move the file, but when you compress them, it's easy and you are more sure that you will not lose your file because of the copying or moving. So to do that, let's just use the gzip to copy. I just want to zip the first one and let's just zip the second one, R2. Yeah, I think they're supposed to be zipped now. Let's do zip. Yeah, voila. Now we have that JZ for all of them. So we can do the fastp for the single end words now. So to do that, we need those parameters or those flags. How to know those flags? You can just go for fastp dash dash help. This is where you have the flag. You can read and go carefully and read to see what you want to use. It's a bunch of stuff, but if you go for the option with dash I, this is for the input one, the read input file name, dash O, the lowercase, this is output one, dash I, this is for the when you working with per end, you need the capital I for the input two, capital O for the output two. So you can go ahead and use it and try to find what kind of flag that you will need based on your analysis purpose. Let's clear again to get more space. Yeah, something you can, if you want, you can also check what version of fastp you are using. Fastp dash dash version. I forgot to tell you that at the beginning. I'm using version 1.1.0. That's good. Okay. So now let's run the fastp for the single end. Let's just follow in the screen. Fastp. Let's just break it like this. Dash I R1, sorry, that JC. My output, it will be, you can clean the output however you want. It's just the output file, clean sample, that FASTQ, that JC. I want it to be zipped also. And the how many CPU do you want? The dash thread. I want 4 CPU. I said, okay, I want 4 CPU. In that case, if you are running with big set of data, you can go up to 4 CPU. That will be faster for you. And you can also go for the qualified quality thread. You can just use dash Q for this part. It will give you the same, and you just precise how many, what is the minimum for quality you want. Let's say we want Q20 in that case. And for the length, the same length required. What is the minimum length required? Let's say I need 50 in that case, 50 BP base pair. So you can just put dash L, it should give you the same result. And now it's time to generate the report. Or if you want to add more flags or more argument, you can keep adding based on the help. It's based on your analysis purpose. I'm just keeping this as simple as possible, as this is just a tutorial. In dash H, this is for the HTML report. Let's call it report or single end report dot HTML. Okay. And for the, if you want also a JSON report, just dash J single end report dot JSON. I think I mistyped JSON. And that's supposed to work. Let's try. If not, we will just troubleshoot to see if I mistyped something. Okay, now it's working. It's detecting adapter sequencing forward one, no adapter. Now I think it should be okay. It worked. It takes about 8 seconds. It's depending on your computer performance. So it just gives you a report in the command line. You can see everything. With one, before filtering, total with 9, and after filtering, total 8, and the filtering result, 8 with past the filters. We have 1 failed due to too short, and you will have the same also if you go and if you go to see the HTML report that's generated. Let me just look it for you. I hope you can still see that. This is the single HTML report. This is the report that is generated. It's like a nice report with a lot of graphic. This is the filtering statistic. This is the before filtering, after filtering. It's just a nice report. It just gives you kind of a summary in the comment line. Yeah, you can go and do based on your analysis. If you go down here, this is exactly the script that you run for the to run the FASTP. That's been with if you send this report, this file, the HTML to someone, it can be able to run the same the same FASTP and get the same result if the person has the same data that you use. It also gives you the FASTP version and when we did the analysis. Okay, let me go back. Okay, let me go back to my terminal. Okay, this is pretty easy for the single end. It's pretty much it. Let's do the per end sequencing. For the per end, what do we need? It's okay. Let me clear my terminal. Yeah, for the per end, we need the input one, input two, output one, output two. This is just the difference. But the scope is basically the same. Let's go fast P. Okay, dash I, capital. Let's call it sample R1 dot fastq dot jz dash I. It doesn't matter if you put the INO. You wanna, it doesn't matter where you place them, but you need to respect the capital and lowercase. Sample that we do that fastq that jc. Now for the output, let's call it clean R1 that fastq that jz. A clean R2 that fastq that jz. Okay, now we can go to detect the adapter for parent. If you want to add that, this is all of those come from the help that I showed you before. Dash dash detect adapter for parent. Or it stands for parent. And if you want the correction to be done, sorry, dash dash correction. And as I mentioned, you can put qualified a word, something like this, that as we did before, qualified quality word, or just dash. You can put dash dash qualified qualified quality word, or just put just one dash dash Q, and I need Q20 in that case, and dash L is the same for the length, L 50, 50 minimum 50 base pair, and dash W, this is for the memory, for the CPU. I need 4 CPU, I said in this case, 4. Yeah, we did need space before the forward slash. And now let's generate the report that H for the, let's call it report that HTML that J that I could report that JSON. I think if I didn't mistype anything, it should work. If I did, I will go back and try to troubleshoot where I mistype. Yeah, I think fail to open file sample R1. Yeah, I mistyped something. Sample R1 that, okay. Let me see. Fail to open file sample. Okay. Okay, I see what happened, guys. It's because I'm just putting the input file that I don't have. So let's try to, we can use just the first one because I renamed the script, but I didn't rename, because I still have in my directory R2 and R1. That's why it failed. Because I just said I have the input, simple R1, simple R2, but it's just R1 and R2. Let's try. If it failed again, I will try to troubleshoot. This is a way to troubleshoot because scripting is not easy. You still need to mess up sometime, but what is important is just to know when you mess up where to go to correct your mess. Yeah, it's working. Okay, it's working. Okay, I done in 7 seconds. That's a good speed, I guess. You will see the same report. It will give you no adapter detected for read one, no adapter for read two. That's good. So read one before filtering nine read pass, and read one after filtering 8. Read two before filtering 9. Read two after filtering 8. And the filtering result, you have 16 read past the filter, and two failed due to too short. Read corrected by overlap analysis. 7 read as corrected. This is what I put the correction. You see the dash correction. This is for the overlap analysis. Base corrected by overlap analysis. 14 base correct for overlap analysis, and 7 read corrected by overlap analysis. You see the duplication rate is 55%, and the size peak is about 187. It's the same if we go to the file manager, you will see let's refresh. This is the report that we generated. It's a nice report. It's also interactive. You see, it's very different than the first one. Yeah, it's different than the first one. It's interactive. You can click if I want only with one, or if I want all of one. If I want, okay. It's pretty interactive. It's based on what you want. Okay, so let's say, okay, I just want for with one and both of them, it just gives you the read one. If you want for all of the reads, it gives you, and there's other stuff you can do based on what you are doing. It also gives you the before filtering analysis and after filtering analysis. It's pretty much the same we did in the single end alignment. Okay. What else we can do? So the adapter, this is just what the adapter trimming do, or just the FASTP can auto detect up to 1 million reads, and the command, like when you run the FASTP, but you can also do the manual specification if you know what you want to detect, when you know the adapter from your kit documentation, or if you already know that you don't have adapter, or your adapter were already removed, you can disable the adapter for trimming. You just use this flag in the command line -- disable adapter trimming. That will disable the adapter. You will not have the adapter analysis. So for the quality filtering, it's just a brief thing. When you use for quality, the Q10, that means you need 90% accuracy. When you use the Q20, that means you need 99% accuracy. Q30 is 99.98% accuracy. Generally, they require a, a quality control that is over or not less than Q20. That means at least 99% of accuracy. You can also use some other quality control, is the sliding windows, the cut right, or cut right mean quality. Cut right mean you go from the trim side, and you can also do the poly trimming. Those are all flags that you can keep adding in your script. If for the length you put -L, that means keep width that is less than, or no, greater, that is 50 or greater than 50 base pair. Is -- length limit 0, that means maximum, there is no limit. what there is no limit for the length. So this is a complete example. This is a complete example. If you want to do, let's just do this complete example, and before we move forward, it's almost the same. Let me just come back to the same script that we have. What changed, and this one you just have the trimming poly G and the cut right. This is just what has added in this FASTP analysis. Yeah, I think you can add it here. I just add dash dash trim poly G, trim underscore poly G, and dash dash cut right. So if you run that, it will overwrite the first report that you generated. But if you want to generate another report, you can just rename the report. Let's say, let's call it updated report. And the same for it. It just up to you, updated. If you want to compare both of them, you just change the report name. I think it should work. Let's see, cross fingers. Yes, it did work. You have, it tell you almost the same thing. You say it's completely different. Let's try to open them and see. Let's refresh that. Uh, I think I probably did something wrong. I have the updated report for the JSON. I don't have it for the HTML. I probably did something wrong. Let me see. Updated report. So it might be something with my computer speed because it worked for the JSON. It worked for the JSON because I changed the name. I have the updated report for JSON. It should also work for the HTML. You can try it to see. You can try it. Maybe it worked for you. Maybe it's something related to my computer. But when I double check the script, I didn't do nothing wrong. It's just the same report.html. I think it just, okay, I think I see what I did wrong. I'm sorry. I think I removed the dash H that's meant for HTML. Now it should work. Okay, yeah, it should work. Now it's say, okay, HTML report generated. Let's just give a quick look. Voila. I got it. Let's see. Let me try to put them in different screens so you can guys see them. Yeah, this is the updated report. And I'm sorry, let me just, yeah, this is the first report. You will see kind of the difference. Let's go to the graph, maybe for visual effect. You see, is there any difference? Yes, you can see the difference. And here, it is easier to see the difference. That's when you put more options, you will probably have different result based on what you are looking for. You see, it's different. Okay, let me just, okay. So we generate that one. What is this command do is remove the adapter sequence automatically, filters low quality base using that is less than Q20, discard with short than 50 base pair. The trim poly G tells that remove the fake G artifact first, the cut right that catch any remaining low quality bases, and also look for the overlap base error correction, generate HTML JSON. Yeah, this is what we use. Okay, this is another way you can do the same thing, but I will not teach you how to write this script. I already have it. I will just show you the script and just close this tutorial, guys. Because when you are working, you can also use a script that do the job for you. And this directory, let me just clear first. And this directory, you can see I have a script that is doing the same job that you have. That is batch processing.sh. Let's just open it. Nano batch processing. You will see what is it's almost the same because I'm working in the same directory. I decided in the script to make another directory to not override the directory that I have already created. It will make a directory that goes fastp class and a directory named raw data, raw for the raw data. It will Okay, it will also make another directory named fastp output. This is just to store the output data because it is not recommended to overwrite the raw data. You need to store the raw data in specific directory and the output in specific directory. So this script will do the same and it will navigate and download the data and do the same exactly this what I did in the command line. This is what this script will do also. Okay, what we can do, let's try to run it and try to see if it will work. Because I'm in this directory, if I run it inside this directory, it might be complicated. Let's try to go home. City home. Now, I know my script name. Okay, let's run it. I will go, it is fast. Sorry. Fast P. It's just the where the script is and the script name. It should work. This is in the home fastp. It is in fastp tutorial. And the script name is batch, something like this. And if I run that, it should create the directory that I mentioned in the script and do the same analysis. Let's try to see. I hope it doesn't fail because I didn't test that before the tutorial. Okay, it's cloning, it's working. Cross fingers. Voila, it worked. That's nice. Okay. Yeah, I have, you see the same analysis. It's just the first analysis. If you want, you can update the script. If you want to update the script with this one, it will, because this script, I think I have it only for this part, the per end analysis. No, I think it's for the combined one. Yes, yeah, I didn't add the poly G trim and the cut right. If you want to update it, you can decide to go ahead and update it. And you can see it worked. That means everything created. Let's go. The first directory created should be the, let's just ls to see. Yeah, ls, I think it should with the fastp class. cd fastp class, fastp I mistype. Yeah, inside this fastp class, you have yeah, the fastp, you have the output and the raw data. This is where we will find the report everything. So before closing, let me just tell you that after you do the fastp, what you need to do next, you are ready for the downstream analysis. So you can do RNA-seq, a variant calling, genome assembly, and metagenomics. It's based on what you have planned for your project. And further reading, I do recommend to read this paper that is Chance and et al. In 2018. This is the original documentation for FASTP. They also have an official GitHub repository. That is where I retrieve the data. This is the OpenGene. This is the GitHub repository. You know, I leave the link in this video. You can also try to read. This is some documentation you can read about multi-QC, NC-core, and FastQC. I think that's it. We've been used FastP for quality control and pre-processing of sequencing data. That was a complete tutorial. I hope it was useful. I hope it will serve you. And if you need anything else, I think I might be posting other video for other bioinformatic tools. That's pretty much it. That's what we have for this tutorial.